Can you take some pictures? So we just wait a few minutes. Yeah. I don't need this, I think. Yeah. Wait, no, they are. It's maybe for them. Yeah, okay.
For those of you who are here for the workshop, just to let you know, this is a regular staff talk that we're just getting into. And afterwards, we're going to go up to the area for lunch.
Hey. Hey, Jay.
Hello.
Nice to finally meet you in person.
Likewise.
Thanks for coming.
You're welcome.
I hope you like the place and the workshop. Yeah. Welcome back.
I learned that long ago.
How was your morning drive here? Yeah. Do you live close to UCLA? Okay. Oh, so you don't even have to take 405. Cool. Okay, sure.
My honor to introduce to you Praveen Billy. He got his bachelor's degree from IIT in 2003 in Double E, and then went to Boston University. He got a master's in physics in the neural system in 2009. And then he was a postdoc and a research assistant professor there for 4 years. Became day girl in 2013. Now a senior member of the technical staff. He's led a number of big projects, including the ones he's going to talk about today, which are DARPA RAM and DARPA RAM replay. And so go to it. Go ahead.
Thank you, Mike. I hope everyone can hear me. Is that right? Okay, good afternoon, guys. Thanks for coming. For the people who are attending the workshop, I hope you are enjoying your day. And for all the people from HRL who are attending for this my talk, I'm very honored that you are here. So before I begin, I do want to take a chance to thank all my collaborators. So over the last 3 years, I have been very fortunate to have DARPA funding. And we have collaborators across multiple universities and companies. And whatever I'm presenting to you today is just a sampling of the things that I thought I could fit into this talk. So there are many works that I'm not able to talk about. So, but the people highlighted in green are present here. You are welcome to talk to them. So as the title shows, I'm going to be telling you about the advances we have made in non-invasively modulating memory formation and consolidation. So 3 years back, when we got this money, the prevailing view in the field was that at least for memory processing, it is very difficult to do a very reliable job at modulating non-invasively. In fact, our funding agency, DARPA, also believed in that view, but they were willing to give us some money to test our hypotheses. So my view was that unlike other brain functions, memory as a process is very distributed. It does not depend on one region. It depends on coordination across the whole brain between the cortex and the hippocampus. Unless there is a problem in the hippocampus, maybe we don't need to intervene invasively. So this is the overview of my talk. So before moving forward, I do want to share with you what I think is the state of the non-invasive stimulation research. And then I want to talk about the experiments that we did in non-human primates with our collaborators, where we directly compared invasive versus non-invasive stimulation to modulate brain function and as well as enhance memory function. We did some fundamental work there, and then we leveraged those insights in the next program that we won, wherein we are developing non-invasive stimulation technology to help healthy humans, particularly improving general memory function as well as sleep quality, and also to target specific memories using non-invasive methods. So we did make some progress, and I'm very excited to share with you on what we have found so far. So given the skepticism in the field about non-invasive stimulation, if indeed these results that I'm going to show you are accepted as time goes by, then indeed we are in a phase wherein non-invasive stimulation technology can be a game changer, can be widely adopted because it works reliably, because we know how to optimize it and make it work for every single user. Compared to invasive stimulation, of course, there's a lot of reduced cost, and there is low risk. And we can also stimulate multiple sites as needed as the person or the user or the subject improves. OK. So I'm sure all the people here are aware of non-invasive stimulation, transcranial current stimulation. It's a buzzword that I'm sure you may have heard of. There are people, hobbyists, do-it-yourself people who have been trying this, and they have blogs where they share what they experience and what sort of improvements they get. There are some companies that try to make money out of these initial excitement about these devices. And I mean, there is one company focused on gamers, and then there is Halo Neuroscience that is selling these TCS gadgets for elite athletes to improve their performance by a few milliseconds to get their gold medal. And then there is this company, Think, that claims that we can use these devices to improve sleep and treat stress, anxiety, and whatnot. As you may know, some of these companies have already gone bust because the initial excitement has faded, and then the claims that they were making were not well supported. But nonetheless, in the scientific community, there is a lot of interest in this particular technique. So today, this morning, on PubMed, I just searched transcranial stimulation, current stimulation, and there were more than 15,000 articles on claiming benefits for various things, ranging from pain to anxiety to even socialization. But there was also this paper that came out, I would say, 2 years back, that did a meta-analysis of many of these kinds of papers and showed that maybe the studies were underpowered, and maybe there is never a reliable statistical effect of this non-invasive stimulation. And last year at CNS conference, Bouzouki made a big splash in the news where he reported on his experiments with cadaver heads, wherein he was trying to figure out how much current needs to be applied to the scalp so that he can elicit spikes from neurons. And he figured out that it has to be in excess of close to upwards of 5 milliamps, at which point the current is no longer safe. So his claim was that unless you are able to elicit action potentials from these currents, you cannot reliably affect behavior. And so this is what is happening in the field. And if you look at many of these papers, what they do is that they have a task, and they are stimulating your prefrontal cortex or targeting areas that are relevant for the task. Luckily, if they get some behavioral improvement, write a paper. And if you don't, then forget about it. That's what is happening in the field right now. So none of this really is governed by our understanding of how the brain works. And because of which, unfortunately, the non-invasive stimulation field as a whole has not been getting the acceptance that it deserves. So in this talk, my endeavor is to present to you results which show that we can systematically modulate memory function. And I hope you will see that there are better ways of doing things like this. So in our first study that DARPA funded, we really wanted to focus on what mechanism underlies these improvements that have been reported in so many innumerable papers. So we implanted non-human primates with Utah arrays in prefrontal cortex and the inferotemporal cortex. And also we had depth arrays in hippocampus. We wanted to record single neurons and local field potentials as the monkeys were getting stimulated non-invasively to try to understand exactly what could be happening in the brain when the monkeys are improving on a task. Since there is a lot of hardware on the head of the monkey and also inside the head post, the pedestals, the wire bundles, the arrays, we really had to develop a very complex finite element model to determine how the current propagates through the head of these specific monkeys, and then optimize the electrode placements on the scalp so that we could be targeting one of the 3 locations or maybe pairs of those locations. And as a result, for the first time, we have in vivo data in response to non-invasive stimulation as well as invasive stimulation. In this talk, I'll be focusing on non-invasive stimulation, but we do have data on invasive stimulation as well. So the first thing we did was we wanted to characterize how the brain responds to invasive stimulation as well as non-invasive stimulation. So we had various parameters. So the paper from Nantia was a starting point for us. So some of the parameters that she used was definitely part of the range that we looked at. At the same time, we also swept parameters for the non-invasive stimulation. So things like how long is the duration, what is the frequency if you're applying alternating current, what is the amplitude of the current. Also, when we optimize for these non-invasive stimulation montages, you could be optimizing for max intensity, which is the maximal electric field at that particular location that you're interested in. Or you could try to optimize for the focality of the stimulation, especially if you're using a high definition montage. We could also try different polarities and different stimulation targets. So what we found was that in this table, the first column is known biomarkers of strong memory formation. So what it means is that in the studies done with primates and humans, what they found was that these biomarkers correlated with strong memory formation. So any memory that was later recalled successfully had that biomarker up high, or whatever the direction may be. So definitely with invasive stimulation, we can elicit those biomarkers. So that's the column for the third column. But interestingly, even non-invasively, we can elicit similar or even better biomarkers to ensure that the memory formation is strong enough. So I'm not going into a lot of details here, but I just want to give you a sense that before this point, the doubt that scientists had on what can we do non-invasively, we wanted to get some clarity on it. What can invasive do that non-invasive cannot do? And what can non-invasive do that invasive cannot do? So we wanted to get some understanding, and this is our first main observation. But then, so in this table, if you see on the fourth column, most of the best montages for eliciting these biomarkers happen to be targeting the PFC. So that's not that's surprising, because PFC as an area interacts with multiple cortical areas, and has been linked to a memory function in numerous ways. And also many of these papers that I talked about directly target the PFC to get those improvements that they write in their papers. So for this particular study, by the way, this paper is going to come out tomorrow, so there's an advertisement for it as well. So in this paper, we targeted the PFC in our monkeys, and we trained the monkeys on an associative learning task. So real quick, the monkeys see images, and they have to forage for a location where they get a reward. So the monkeys are not told where the location is, but so initially, so the scan path in the very first presentation, the monkey has no idea, so it spends a lot of time. But then as the memory forms and is encoded, then the next time it sees the image, it knows exactly where to go. So we also had some non-behavioral data collection blocks, wherein we're just stimulating the brain and trying to understand the differences between the TDCS, the transcranial direct current stimulation of PFC, and sham stimulation, how they differ. So if you have any questions, please feel free to stop me, because there's a lot of stuff that I'll be covering, and some of it may be very neuroscientific. So if something is not clear, please raise your hand. So this is the behavioral result. Like all the other papers, we did find that non-invasive stimulation improves performance. So this is performance from a single TDCS block. So as you can see for a particular image, as you can see initially, the reaction time is high, close to 15 seconds. And with every presentation, the reaction time drops, and it flattens out. So there is a nice sigmoidal behavior to it. So we fit logistic functions, and we extracted two critical parameters, the N50 parameter and the offset. So the offset determines the final behavior. And the N50 parameter determines how many trials you would need to come up to some criterion performance. So what we found was that with the TDCS and sham, the final performance, or the offset, or the min RT, is not different. And that tells something about that tells us that stimulation is not enhancing arousal levels or enhancing attention, or it is not enhancing your ability to execute the motion, the movement. It is doing something else. But more importantly, there is a clear difference in the N50 parameter. So that means this curve shifts leftward. In fact, the learning speed is accelerated by about 40% because of this stimulation. So this is interesting, but not novel, because many people have already claimed similar things like this on not just this task, but on many other tasks. So the next thing that we looked at was the single units and the multi-units. What we found was that, just as Bouzouki said, for 2 milliamps current, the single units don't elicit more spikes. That's confirmed. So as you can see, for PFC and ITC, the firing rate of the single units is not different between the two conditions, as well as for the multi-unit activity. And we also looked at the selectivity of these single units and multi-units to classify the images that are shown in each block using an LDA procedure. And even here, there was a drop in selectivity for the multi-unit activity, but it was not statistically significant. In general, the behavior of the single units is not changing because of this stimulation, but we are still seeing improvements in behavior. So what it means is that probably behavior does not always depend on the firing of single units. Maybe it depends on the timing of the single units without affecting the firing rate, or maybe it is dependent on other coordination-based mechanisms like coherence or synchrony. Another thing that we noticed was that if we are targeting the PFC, you can see how the local field potential or the local network activity changes from the sham condition to the active condition. So you see this very large oscillatory activity in the low frequency bands, especially. But in the ITC, which is in the temporal cortex, very distant from the target of the stimulation, we don't see any changes. So again, in the field, people would say that non-linear stimulation is very distributed, widespread, and you can't possibly have any local effects. Let's finish one point. So similarly, if you're targeting the ITC, we see some local changes in ITC, but we don't see any changes whatsoever in the PFC. Sure.
Could you just briefly describe what TDCS is doing to the brain?
Sure. So we are injecting current. It's being shunted by the skin, the skull, hair, whatever. I mean, the musculature, and whatever current is able to go into the head will be some sort of an electric field that's operating in the extracellular medium of the cells. So the mechanism that we think is that it is depolarizing or hyperpolarizing the membrane potential dynamics of cells at a level that it is not eliciting new spikes. So it is very sub-threshold modulation of the membrane potential dynamics. Sure.
LFP for prefrontal cortex, is it normal that it's that low in amplitude compared to the ITC?
I'm not sure about the scale.
When we're looking at the sham.
I'm not sure how to answer that. I don't know. So the PFC is very sparse. Sparser than ITC, because ITC is responding to visual objects, shapes, and categories. So there is something always active, but PFC is a very heterogeneous structure. So it also depends on from which part of the PFC we are recording from. But I don't have a good sense of why that amplitude is so low.
How many monkeys are we seeing data from?
2 monkeys.
And so are they consistent?
Yes,
Or
yeah.
How do we interpret the error bars in relation to that fact?
So here in this plot, the data is combined across the monkeys, between the monkeys, I should say. But I'll show you a plot where we are seeing the data separately for the 2 monkeys. And we have corroborated all the findings individually in each monkeys. And all the statistical analysis that we do, we combine all the data, and then we have the monkey identity as a covariate in the analysis. And that never comes out as a significant factor. But I'll show you some specific data. OK, actually, right here. So, so here, the y-axis is the change in the LFP power in the different frequency bands. And the y-axis is being plotted in log scale because of the range that we are seeing. So there is a significant, and the 2 colors are the monkeys. And the statistical test is corrected for multiple comparisons. So is the enhancement compared to the baseline significant or not? So this, when we are targeting the PFC, we do see a very broadband increase in spectral power in all bands in PFC, but not in ITC. So there is some change, but it's not significant at all. And you can compare the ranges. We also looked at the spike field coherence. So here we looked at, in particular, we looked at the synchrony between the LFP in one area and the multi-unit activity in the other area. And what we find is that, in general, in the high-frequency bands, there is no change. But in the low-frequency bands, there is a decoupling, significant decoupling in the delta and the alpha bands. OK. Within the PFC, we did find, where we are targeting the PFC, there is, within area coherence, increased because of the stimulation. But again, in ITC, which was not the target, we did not see any significant changes in coherence within the area. But most importantly, what we found was that the inter-areal coherence, which is the coherence in the between the LFPs, between PFC and ITC, showed some weird, not so weird, but initially we thought it was weird, but now it is being matched with data from the invasive stimulation groups. So what we find is that in the low frequency bands, the communication is impaired between PFC and ITC. But in the high frequency bands, like the high gamma and the low gamma, we see an improvement in the coherence. So, so far what I have shown you is that when we are targeting the PFC, we find significant neural changes in within LFP power in the PFC. And we also see a broadband increase in the coherence within PFC. And we see a decrease in the spike field coherence in the low frequency bands. And we see a decrease in the interareal coherence in low frequency bands and an increase in the coherence between the brain areas in the high frequency bands. So we tried to understand which of these significant neural changes correlate with behavior. And we had a very elaborate process to determine that. But at the end of it, what we found was that only the changes in the interareal coherence that are induced by the non-invasive stimulation can predict the improvements in the learning speed that we see in our non-human primate subjects. So for the first time, at least we have a mechanism by which the non-invasive stimulation could be enhancing performance on a learning task. Interestingly, the UPenn group that's also being funded in this program, they are also seeing this sort of behavior as a biomarker for strong memory formation in their epileptic human subjects, which is a decreased coherence in the low frequency bands and an increased coherence in the high frequency bands. So to drive that point into your heads, what I'm saying is that the biomarker for memory formation seems to be common between non-invasive and invasive, and it can also be achieved non-invasively. I don't have to summarize this, but I already made these points. Moving forward, sure.
What was the stimulation frequency you're using again?
This was direct current, so yeah. Yeah, so it ramps up from 0 milliamps to 2 milliamps in 3 seconds. It stays like that until the block ends, and then there's a ramp down for 3 seconds. But we also collected neural data in response to TACS. And we are still crunching the analyses, and we will have lots of papers on those. I'll be happy to share you what we have on that. I mean, there is one hypothesis that maybe when you're applying alternating current stimulation to the brain, you are entraining oscillations in different areas. That's not true. And we also have recordings from the hippocampus, which I'll be showing in a short while. So even we can get modulation in a deep brain structure non-invasively. So the standard in the field is to just turn on the stimulator for tens of minutes or even an hour while the subject is performing the task. So we wanted to figure out if maybe we can use very short duration pulses of this non-invasive stimulation. There's an illustration here. So when you're involved in a task, maybe the critical periods where you're getting relevant information is very brief. So why don't we just stimulate during that particular duration only and not just stimulate when you're idling or not receiving any relevant information? So for that, we have to, so we proposed and got funding to start looking at what happens in the brain when we stimulate it with pulses as short as 400 milliseconds. So I don't have the behavioral data for this one, but I do have statistically significant neural effects as a result of the TCS pulses. Actually, here's an example. So this is a recording in the hippocampus in response to stimulation at ITC. So and it was just 400 milliseconds. So here we are looking at the power in some particular band, in this particular band, yeah. So you can see how the power increases as a result of this pulse. We're not looking at data for 5 seconds after the pulse because of the artifacts, but you can see that there is a lingering effect of the enhancement in in that beta band or whatever in the hippocampus as a result of non-invasive stimulation of ITC. Similarly, the coherence between PFC and ITC can also be enhanced with 500 milliseconds of stimulation. Just imagine 0.5 seconds of non-invasive stimulation. But there is an effect on the brain. And we have exploited this tremendously in our experiments with healthy humans. I'll show that to you as we move along. So we built a model wherein we are trying to predict whether there is going to be a significant modulation or not, and the magnitude as well, in response to all the stimulation parameters, and also using the current brain state. And this was directly inspired by the work from Kahana's group within the program, where they showed that if you stimulate the brain when the brain is in a good encoding state, then you actually impair memory function. So their mantra is stimulate only when the brain is in a bad encoding state. So that means if you are not stimulating in closed loop with the brain state, then you actually may be impairing performance. And that's exactly what they showed in their neuron paper. But then they followed it up with this closed loop paper in current biology, where they showed that indeed there is an improvement when you are cognizant of the brain state. So, so what we found was that for the PFC modulation, again, we have these biomarkers. We figured out the relevant factors and the interactions on how those affect the modulation. So very clearly, we can see that the pre-stim brain state has a negative effect on the biomarkers. And we can see that here in this plot. So on the x-axis is the pre-stim power. So in this case, it's the theta power in PFC. And on the y-axis is the modulation index. So if the pre-stim power is high, which will correlate with a strong memory formation, and if you intervene, you actually get suppression. But if the pre-stimulation power is low, you actually get enhancement. So again, whatever was true for invasive stimulation is most certainly also true for non-invasive stimulation. And here are some of the best montages in whatever we have recorded in terms of eliciting these biomarkers. So as you can see, for this particular montage and parameters, with just 500 milliseconds of stimulation, we can modulate the theta power in the prefrontal cortex. And we can also get a decrease under certain conditions. And we can also increase the gamma power in the PFC. So similarly, for hippocampal modulation as well, we found that the pre-stim has a reliable negative effect on the biomarker. And interestingly, what we find is that if you want to get maximal modulation in the hippocampus, then you should be using the montage that was optimized to target the location of the hippocampus. And again, here you can see how examples of this modulation in this table. Similarly, for synchronization, again, the pre-stim coherence has a negative effect. So one thing we noticed real quick is that if you are trying to enhance the synchrony between two brain areas, it's always a good option to target at least one of them. You get the most effect when you target both at the same time. But if you're targeting non-interested areas, then you are not going to see the biggest benefit. So just to summarize this experiment, for the very first time, we showed that even with very short pulses, you can get significant neural modulation in the biomarkers of interest. And the ongoing brain state is also an important factor for the effects of the pulse TCS. So we better be focusing on that if we want to develop closed-loop applications. As I showed you in the first experiment, the effects of the stimulation are maximal at the targeted area, and the synchrony effects are maximal when you are at least targeting one of the areas of interest. And another thing which is very clear is that just merely increasing the current amplitude, jacking up the current, does not give you the biomarkers that you're interested in. So brain is a very complex nonlinear machine, physical substrate, so it can't be so simple as that. So we have a lot of data to show that that's not the case. So anyway, for this field to be very successful and reliable, we need to be using pulse TCS, which is timed to the learning event, and that should be dependent on ongoing brain state. If you don't do that, then the likelihood of impairment of your memory function is very high. Okay, so just switching gears. So I'll be presenting 2 experiments where we leverage these insights that we got from the non-human primates into developing a man-portable system that could be used by healthy humans to accelerate their skill acquisition and learning performance. So our metric was we had to show improved retention over 2 days compared to the state of the art. And we had to demonstrate these improvements in ecologically realistic tasks, not the simple toy tasks or academic tasks. So we worked with USC ICT to come up with these immersive virtual reality environments to assess the memory performance. And we proposed to be the first to use high-density distributed non-invasive stimulation patterns to tag specific memories and also cue them during sleep to enhance performance on those particular memories. Our goal has always been and still remains to make this into a commercial product, but one that is based on results and one that is based on optimized understanding of how the brain encodes and consolidates memories. So this is all about sleep. So whatever I have showed you so far has been intervention during learning. But as you know, if you don't remember what you did maybe last Wednesday, that probably means that you have not consolidated those memories well enough. In fact, the brain has a difficult task of deciding which memories to replay. So in effect, the things that you consider are important may not be the ones that the memory system may consider to be important. So nonetheless, there are existing studies which intervene during sleep and showed some improvements. So all of them try to deal with forgetting. So you're definitely going to forget, but how much can we stop the forgetting is the deal. You may have heard about the olfactory cueing, the work that came out in the 2000s from Germany, wherein when the subjects are learning a simple paid associates task, there are some aromas in the air, and then while they are sleeping, those aromas for a subset of the cues were presented, and then there was some improvement for the cued memories. So this was also replicated by Ken Paller I think in 2007, where he used auditory cues. So pairing when the subjects are learning some associations between an object and a location, some semantically relevant auditory cues played, and he showed some cueing benefits with auditory cues as well. And then our collaborator, Lisa Marshall, she used open loop TACS to boost slow wave sleep during non-REM sleep, and she had some improvements as well. But the problem with all these studies has been that the demonstrations have been on very simple tasks, not ecologically realistic tasks. And the sensory cueing methodology is very interesting, but maybe impractical for operational use. So just imagine if you are in your son's wedding, or if you're in a rock concert, and you really want to remember that memory with all the noise and everything that's going on. And say if you play some cue, auditory cue or some olfactory cue, and hoping that it's going to be associated with the event, it's probably not going to work. And also, the number of discriminable sensory cues is limited. And these cues, as I said, are directly competing with all the ambient environmental cues. And in fact, the auditory cues may actually disturb your sleep if the intensity is high enough. If it's too low, then it's not going to work. And so finding that sweet spot is, again, a big deal. Nonetheless, this is what we proposed as something along the lines of the targeted memory reactivation literature, but doing it completely electrically. So we proposed a two-pronged approach wherein we are intervening during waking as well as during sleep. So during waking, our proposal was that we are going to be stimulating the prefrontal cortex. So based on our monkey results, we know that stimulation of the prefrontal cortex improves your learning speed. So that means whatever you're encoding or whatever memory you're forming is going to be done more efficiently and strongly, which itself can lead to those memories to be replayed to a greater extent than the other ones during when you sleep. On top of that, we came up with this idea called STAMP. So STAMP stands for Spatiotemporal Amplitude Modulated Patterns of Transcranial Current Stimulation currents that are being applied to tag a particular learning event. So for any learning event, you can just come up with some STAMP pattern. It could be a spatial pattern, or it could be a temporal pattern. Whatever that is, once you apply it during the learning event, just like the auditory and the olfactory cues, you could be using them during sleep to probably enhance the probability with which those memories could be replayed, and then you could see improvements in performance. So the advantage of STAMPs is that the diversity and the variety of the cues that you can come up with is just huge. There is no competition with environment. In fact, many of our subjects, they can't differentiate whether they're getting stimulated or not during the night. Some of the subjects, so we recently tried to do this live in a hotel room at some program review, and our STIM subject, or actually participant, claimed that he was a sham subject, sham participant. So it was, once they get into slow wave sleep and once you initiate this procedure, the likelihood of them waking up from that is very low, unless they are a very light sleeper. So during the sleep, so the replays are hypothesized to happen during the stage 3 and 4 of non-REM sleep. The duration and the power of the slow wave oscillations has been correlated with memory performance. So in a normal person, these slow wave oscillations are not robust. I mean, the brain goes in and out, even though you may have a very prolonged duration of slow wave sleep. If you look at the oscillations, they are not uniformly strong, let me put it that way. So we proposed a closed loop TACS method, which would lock to the phase and frequency of the oscillations and then apply them just to keep the rhythm going. And then once you have an enhanced slow wave sleep real estate, then you can now start stimulating with the stamps of the memories that you're interested in on the positive half waves of the enhanced slow wave oscillations. So here, during waking, the learning event could be very long. It could be on the order of like tens of seconds. It could be a minute long. But during the night, you only have about 0.5 to 1 second to stimulate. So it has to become very shortened. But that's what we do. So I'm running out of time, so let me go real quick on the experiments. So in this task, we first wanted to prove the efficacy of the closed loop TACS. We had a thread detection task. So here there is a tripwire. So the subjects have to detect the presence of threats. During training, if they make an erroneous response, they are given a feedback that does not tell them exactly where the threat was. The only feedback is that there was a threat and the subject did not pay attention, so something bad happened. So what we, let me quickly jump into the results. So we had this timeline wherein we do the training during waking and then we intervene during sleep. And then we assess their performance in the changes from before sleep to after sleep based on this closed loop TACS method. So we are looking at a virtual EEG channel centered on the CZ electrode. And we are looking at the last 5 seconds of data to predict where the next upstate could be happening. And we can time our stimulation to be in sync with that predicted upstate. So here is the first result. It's a within subjects design. So in the first week, whatever may be the condition, the subjects are trying to detect threats of a particular category, and in the next week, they try to detect the threats of the other category, and everything is counterbalanced. So here we are looking at the change in the hit rate for threat detection from the baseline test. So this is where the sleep is happening between immediate and morning test. So as you can see, for the stimulation condition, there is no forgetting as such. But for the sham condition, there is forgetting. And when we do it, the paired t-test, we can see that the afternoon performance is significantly different between the stim and sham in their ability to detect threats. So we looked at this more carefully. So what we found was that this improvement is coming from the ability of the stim subjects to perform better on novel images. So the images that were different from those shown in the training. So here I'm plotting the F1 score. So for the repeated images, there is a decrement in performance for all conditions. But for the novel images, there's an improvement for the stim condition, but a decrease for the sham condition. And the same data is being plotted here. So you can see that the largest improvement overnight is coming from, for the novel images, is coming from the stim condition. I don't have time to go into this, but we have developed a pipeline to look at the changes in the spectral power following the stimulation event to see if we can correlate with performance. So in effect, we can extract a spatiotemporal cluster in the scalp EEG, in the EEG that follows the stimulation event, in a reliable way in which we can predict the performance, what the performance would be of the subject, just based on looking at the activity in that cluster. So I can talk to you about this during lunch if you're interested. So we did find a cluster that correlates with behavior that's basically parietal and central, maybe right lateralized. So not only do we have a behavioral effect, but we also are seeing some neural changes caused by the stimulation that we don't see in the sham condition that can explain the changes in the performance, the improvement in the performance for the novel images compared to the improvement in performance for the repeated images. Sorry, I have to go real quick, but we also saw that these subjects show improvements in sleep quality. So this was not a target of our investigation, but nonetheless, all the participants, they reported their sleep quality. We used the Karolinska sleep diary. So what we found was that for all the active nights, the sleep quality assessment was higher compared to the sham condition. We probed this further. So interestingly, what we find is that so the sleep quality. So this is the sleep quality that subjects have in their own rooms before they come to the lab, and then in the acclimation night, their sleep quality drops. So this is like if you stay in a hotel, travel to some place, and so the next day when the subjects receive stimulation, their sleep quality improves. In fact, it's more than, at least qualitatively more than what they have in their home. But for the subjects that don't get stimulated, it's about the same. So what this means is that, and actually the 1 week later when they come back to the lab and they don't have an acclimation night, we do see an improvement for the stim condition, but it is not statistically significant. So the only effect that we have is after an acclimation night, so we can improve your sleep. So we have to optimize this, of course, but this was just a corollary result. So what this means is that if you are staying 2 nights at some new place, then we can help you on the second night. But we will of course try to optimize this and help you on all nights. But Okay, moving on to, so whatever I showed you was, if you intervene during the night, then it will enhance your memory function in general. I mean, it will try to improve your ability to generalize to new images. It has a very generic effect. And also it improves sleep quality. So the subjects report sleeping better. So that's great. But the mandate for our program was to really enhance specific memories. So so I talked to you about the STAM method. So we worked with a company called Neuroelectrics to come up with a library of STAMs. So the idea, so here actually are the STAMs. So we have 32 channels of stimulation, and each column in that image is a STAMP vector. So these STAMP vectors were obtained by optimization based on the orthogonality of the current flow distributions in the brain volume induced by these montages. So we have 256 stamps, and in the experiment that I'm going to present now, which is my last experiment for the day, we are using 14 of these 256, randomly picked. So so this task is called Operational Events Reporting Task. So this was done in virtual reality, developed in collaboration with USC. The subjects are viewing 14 episodes. So each episode is something like this. So it's about a minute long. So something should be happening soon. So there's a person walking to the middle of the building, and then he goes inside. There's another person, a different person, of course, different clothed person who is also walking to the middle of the building, and you can see the other person in the room, and this guy also goes into the building. And then a third person, very different, very particularly different person, is coming, walking, and he stops and maybe is trying to listen in on the conversation, and the audio is not working. They were speaking some Arabic, and then they come out and they are chasing this person. So anyway, so we wanted to come up with a declarative memory task that is very different from the typical paid associates task that is used in the literature. So we created 28 episodes like this. We call them vignettes, and it was a within subjects design. So we divided them into two sets of 14 episodes. So we for the stim condition, we stamp all the episodes, and then only half of them are queued during the night. So so as I mentioned to you before, the stamps are locked to the episode. So so whatever episode you saw, it was about a minute long. So when the episode begins, the particular stamp pattern that we chose for you randomly is applied, and then it stays on until the episode ends. And then there is about a 7 seconds interval, and then the next episode. So yeah, I don't want to go into the timeline, but following this, the subjects have to do a quiz. So remember, they saw 14 episodes. So you guys just saw one episode, right? So try to see if you can answer these questions. So for each question, there is a declarative statement made about the episode. The subjects have to give a true or false response, and also rate their confidence in their assessment of the declarative statement. So some of you may be feeling that it is easy. Some of you may have just forgotten the episode already, or may not remember the details as well. So remember, the subjects have to do this for 14 episodes, and the questions are all mixed up. So for each question, there is a prompt text and some images of the characters in the episode, and from that, they have to try to recall the episode and make an assessment about the statement. So in any way, we use the response and also the confidence from these subjects and build a ROC curve. And the area under the ROC curve is our metric for episodic memory recall. So real quick, so what we show here is the absolute AUC metric, which is the memory metric, over the 3 days. So as you can see, by the third day, there is a significant difference between the episodes that were tagged and queued, and the episodes that were tagged but not queued, and then the episodes that were not, there was no intervention for. So also the tag and queue episodes show the biggest improvement in performance compared to the baseline on day 1. Again, we extracted the sleep EEG biomarkers. So in fact, this one is a very late biomarker. So this starts about 9.9 seconds after the offset of the stimulation. And it lasts maybe about hundreds of milliseconds, 800 milliseconds actually. So the activity in this particular spatiotemporal cluster can predict what the subjects are going to be performing on when tested in the morning. Another thing to note is that since these biomarkers are appearing so late, we realize this very early on. So when we apply the stamp during the night, we wait at least 10 seconds before we can apply the next stamp, just because we don't want to interfere with the cascade of processes that may be triggered by the application of the stamp in the upstate. And that helped us to actually get better results. So that's another insight that we have. Even on night 2, we have a different biomarker that explains the performance. But this one is an early biomarker. So we see all sorts of biomarkers, early, late, but it is not like you apply the cue during the upstate and everything is happening in that upstate. It could trigger processes that could be happening over multiple upstates. So we have to be cognizant of that. Also, real quick, we found that the improvement in confidence for the tag and queue episodes is higher for the tag and queue is higher on the correct trials. And also the reaction time reduces more for the tag and queue episodes. And on the error trials, even though we don't have a significant result, we do find that the confidence reduces on the error trials, and the reaction time increases on error trials for the tag and queue episodes, even though there is no significant effect. So what this means is that if you're getting tagged and queued, as time progresses, your confidence when you're going to get them right is going to be high. And if your memory is weak or you're not confident, then you're most likely to be wrong. So that was interesting to note. Anyway, just to summarize from these 3 studies, and I'll be done with this, is that in healthy humans, we developed the closed-loop slow TACS method that's applied in phase with the ongoing slow wave oscillations, and that helps improve your memory function in general. So if we are not able to remember things, and if you just want to have better memory ability, then this will be more than sufficient. And also, if you feel that you are not able to sleep well, then this will surely help. In fact, it will help me because I'm now 35, and every year I'm thinking that I'm not able to recall things. I'm feeling that right now, and I don't know what will happen when I retire. So we are also able to extract sleep EEG biomarkers that can account for performance changes. So in fact, we have a cross-validation approach where we train the biomarkers on one subset of the subjects, and then we are able to predict the performance on the subjects that we don't use in our training. And we can reliably do that. So that's, I think, a good achievement. And in fact, for specific memory enhancement, in an ecologically realistic declarative memory task, we have demonstrated that we could use the stamp method, which is not sensory queuing, to tag specific episodes and then queue them in a way that improves performance on them when assessed 2 days later. I don't think anyone has even shown this invasively, but as far as I know. Anyway, so with that, I again would like to thank my amazing team spread across the world. And I think we're out of time, but I'll be happy to answer any questions that you may have. Thank you. Sure.
Going back to the beginning where you had that study of the cadavers that says you need too much current. So what of your studies kind of contradicts their result or suggests a limitation of it?
It does not contradict. It actually is in line with them. So in the cadaver, there is no behavior, right? So the only claim that, by the way, the paper is not published, it is just a presentation at a conference that made so much news. The hypothesis was that unless you can change the firing rate of single units, you cannot change behavior. And to get to that level, you have to really jack up the current, at which point it is going to burn your skin. So then people suddenly started questioning if the non-invasive stimulation is actually real or not. Are people just faking or shamming it? So that's what we also find, that for 2 milliamps, which is a safe level of current, we don't see any changes in in the firing rate of the single units, confirming what he found in a behaving animal, actually, where the dynamics of the brain the biochemistry is very different. But nonetheless, we have the same conclusion. But what is important to note is that despite that, we see improvements in behavior. And not only that, we can reliably correlate with certain stimulation-induced neural changes in the brain. So that's the difference.
And I may add, it's a bit funny. So this is vastly different than, just like a human or, live.
Yeah.
Question on the last setup, there was pretty interesting stuff. So you had a unique stamp for each episode.
Each episode, yeah.
So when you say the tab result, is that when you had the same stamp across all episodes?
No, no.
You didn't try that?
No.
You just use a standard pattern across all these episodes and see if that is good.
That may confuse, right? So if you're associating one stamp with everything in the day, then
We don't know.
we don't know. But then for already this task is very difficult. I mean, all our subjects say that it's just very difficult to remember facts about these episodes when you ask them like 1 day or 2 days later. So they're already starting mixing up things in their heads. But despite all the difficulty, we can see a statistically significant improvement in performance for the episodes that were tagged and queued.
So you could just do a cross-tag thing, right? You could present the wrong tag with the cue.
Sure. Yeah. So So exactly. So so during recall, we are not stamping. We're only stamping the experience during learning. But our hypothesis is that if you are getting incongruent stamp during recall, then it will actually impair your performance, and a congruent stamp will improve your performance further.
Okay, so you're not actually giving the stamps during recall, I would think.
No, we're not.
Unfortunately, I didn't pick that up.
No. So sorry. I mean, I could not go into all the details, but we are stamping during learning, and then stamping during sleep. That's it.
You're stamping during learning and sleep.
Yeah. Exactly. That's it.
And so the second question is, do you know which is actually, the effect was present in the immediate recall. So that was my question there.
You mean when you say immediate
Way
recall?
back, I'm not sure
Oh.
about this slide, but maybe it was an earlier study using earlier study.
Sure.
Right there.
Yeah, so here we're not stamping. Here the intervention was that you're stimulating the PFC during waking, and then you're applying closed loop TACS during sleep. So there is nothing specific to any learning stimulus.
Right, but still the effect could mostly be during the waking effect could then be producing the knock-on effect that occurs after.
That's true. Yeah. So we don't have data where we just intervene during the night. I mean, in this particular experiment, we intervened during both waking and sleep. But
Subsequent work you could
sure, yeah,
separate those two things.
yeah, sure.
In particular, the stamping during waking and not during sleep would be interesting. I mean, I recognize that you're helping people sleep.
So actually, the tag episodes, right? They're getting stimulated during waking, but they are not stimulated during the night. So we already have that control. But one thing I do want to say is that we can also look at the changes in performance just during sleep, right? From a test before the sleep to the test in the morning. And we could, we see that the improvements are significant in several measures if you just look at the performance changes overnight. And that should be a result of the intervention during sleep. In fact, we went further and the baselines could be different for the two conditions. We averaged the baselines and then looked at the change in performance from that average baseline. to the morning and afternoon. And despite that, the changes or the improvements are significant for the subjects who got, or actually from the data where the subjects got the stimulation during the night. But yeah, we can always do one where we're just intervening during the night to settle it once for all. Yeah.
I don't know if you're familiar with the DeepMind works on prioritized replays. So they have some experiments where basically they show that replays are not uniform sampling of daily activities, but prediction error and rewards come into play. If I understand, you're sampling uniformly to the stimulation that you give at night. Or do you choose with some, what is the ratio of each one of the different stimulation based on
Yeah, no, that's a good point. So we are actually going towards that. So in our case, we decide experimentally which episodes we want to enhance or cue during the night. So that's something determined by us.
One episode per night? Or?
No, 7 episodes.
Okay.
And we have 7 episodes that are not being cued.
With the same frequency?
With yeah, so we just cycle through the night where we just keep applying these stamps in random order with every cycle. But what we want to be what we are doing right now is there are 2 things, right? One is that if you keep on stamping during the night, maybe you are disrupting the consolidation of other memories that your brain may be interested in or may be relevant for you. So what we want to do is actually that's an ongoing work. We want to predict what the performance is going to be based on the replay right now, or the biomarker at that moment. So then if we keep track of how many stamps we applied and how much improvement we are going to be seeing, at some point we can say this memory has been consolidated. Let's move on to something else. So we could prioritize based on that. And then when all the memories that you're interested in have been consolidated enough based on our computational models, then we just let the brain do its job, in which case it will help the other memories that are not important to the employer or the experimenter or maybe to the well-being of the brain. Yeah.
So have you considered whether the tag memories are actually, the tag and key memories producing interference with the recall of the ones that haven't been tagged and cued? I mean, you might imagine that
Sure.
competitive,
Yeah.
or if you had sort of, like, I don't know if you're familiar with the kinds of studies that Ken Norman does, where you have a cue that has 2 different associates with it, so you kind of like reinforce this learning of one of them that might impair your ability to recall the other one that's associated with the same cue. That would be a sensitive way of getting it.
Sure. So
I
yeah.
mean, it's a question of whether there's upside as well as downside. Too much learning at this might,
Sure.
produce an interference with that. So.
Yeah, no, that's an interesting thing. So right now, the only thing that we have is that we are using the stimulation for, the stamping for all the episodes, and then we are only using a subset of them during the night. So probably we are not interfering in the sense that we are reducing the opportunities for the other memories to replay as much.
Yeah, but the weight changes that occur in learning the ones that are being stamped could also be negatively impacting the weights on the ones that weren't being stamped.
Yeah. Good. Yeah.
So encoding and retrieval are both frontally mediated, whereas consolidation's medial temporal lobe. And so with study 2, you show that the previously shown information actually wasn't improved, but novel information, the reaction to novel information was, which suggests not encode consolidation, but something more frontally mediated, whether it's increase in attention or procedural learning. So I'm just wondering, with these tags and cues, are you providing additional cues to enhance retrieval, which I think is in line with increased confidence, which is highly collinear with increased
we're not intervening. No, during the recall tests, where they are answering in response to those statements, there is no intervention whatsoever. So actually, I think we need to move on to lunch. So I'll be available at lunch. We could continue this discussion because the workshop again resumes at 2, and we just have 45 minutes to eat our food, which is, I think, already set up on the 4th floor terrace.
Where's the press?
Tomorrow.
Where?
Current biology. Thank you. Yeah.